Hello and welcome to Science Research Weekly, Season 4, Episode 30, Antarctic Submarine Canyons. Get ready for, you guessed it, Antarctic Submarine Canyons, but also medicated gel for diabetic wound healing, rapid electronic detection of infectious and chronic diseases, SAS and R in tandem, and several grants for private foundations. This episode covers materials from August 8th to August 14th. Let's get started with our research articles. First, up here, the geomorphometry of Antarctic submarine canyons. Researchers using this International Bathymetric Chart, version 2 of the Southern Oceans, were able to actually detail out 332 Antarctic submarine canyons, and some of these are really big. So they were able to go through and looking at differences between, say, like West Antarctica and the Peninsula and East Antarctica. So really fascinating sort of oceanography, geography, geology. Next up, engineered sEVs encapsulate in GelMA facilitated diabetic wound healing by promoting angiogenesis via targeted thrombospondin 1. So chronic non-healing wounds, that's a real problem in diabetes. Here, researchers were able to use this hydrogel, which had these embedded I think microRNA 221 3P loaded small extracellular vesicles. That's what they call it. So cell biology magic. They loaded them up, applied them to cell culture models of diabetes as well as a diabetic mouse model, and found that they actually improved wound healing and angiogenesis. And our third article for today, Nanoparticle-Supported rapid and electronic detection of SARS-CoV-2 antibodies and antigens at the Subfemtomolar level. So researchers use this gold nanoparticle supported rapid electronic detection, also known as NASRED, this platform that essentially can recognize specific antigens and turn a color, and then you put that in the machine, a pretty inexpensive machine, and it gives a standardized readout, has high specificity, is very sensitive, and it can deliver results in very little time. So using this now NASRED, their researchers were able to distinguish the viral antigens from those of human coronavirus in less than 15 minutes. A great quote for this at the end of the abstract: with low reagent costs and a compact system footprint, this modular digital platform is well suited for accurate near-patient diagnosis and screening of a wide range of infectious disease and chronic diseases. So coronavirus, that was just the test case. It could be used for a lot of different diseases. So very interesting interplay of technology, of chemistry, of biology for medicine and diagnosis. All right, with that, let's switch over to our research tools. Just one thing for you today. From the SAS blog, Emmett Smith's on SAS users, building a seamless data pipeline across multiple languages, a life sciences and healthcare use case leveraging R and SAS. So he used an example of classifying heart disease and showed how to use SAS for the data cleaning and exploration, and then switching over into R to train models, and then also app development. So showing how you can work together with these two platforms. There's no R bloggers posts of note or new cram packages, so we'll jump right into our final section, research funding. And I have 3 research fundings for you today. First is from Gateway for Cancer Research. It funds phase 1 and 2 patient-centered cancer clinical trials with a potential shift paradigm for standard of care. They have a bunch of different programs available, including the Gateway Traditional Grant Program, the Joint NCI SPORE P20 Program, Integrative Research, Joint Society for Integrative Oncology Program, and then the Decentralized Clinical Trial Initiative. Another research opportunity from the Institute for Advanced Food and Nutrition Sciences, or IAFNS. They have from their Cognitive Health Committee this request for proposals. The goal of the project is to identify candidate nutrients or bioactive food compounds to that might support brain health in different life stages. And The recommended approach apparently is a review of systematic reviews, and I guess going from there, the reviews should evaluate nutrients or bioactive components specifically as related to brain health outcomes, not other outcomes, but brain health. That's the big ticket item. And third and final, from the Simmons Foundation, this collaborations in ecology and evolution. And it seeks to create collaborations, strong collaborations, cross-pollination of ideas between investigators, vision statements, that's what they're seeking right now, for new breakthrough areas of ecology and evolution for high-impact funding by the Simmons Foundation. It's interested in, again, idea-generated research and focused on basic principles of ecological and evolutionary dynamics, especially their interface, and might be overlooked. This is a key point. Overlooked or too risky for other funding organizations. So you have wild, crazy, off the walls ecology, evolution ideas. This is your opportunity. They want to engage bold, cutting edge, and innovative research, and then each collaboration will be funded for 5 years with the potential for one renewal. Antarctica or bust. Thanks for joining me for Science Research Weekly. Science on.